everybody. Thank you for and welcome to coming to the Beaver Institute Science and Research Group Speaker Series. Just a couple of quick announcements before we get to our speaker today. For those of you that were able to make it to BeaverCon and came to the open house and stopped by our table, thank you so much. It was really nice to meet people face to face, put some faces to Zoom boxes. And Brian and I are working on setting up our tasks for next year. We will be keeping the speaker series going. In that vein, our next talk in December will be Alexa Whipple. I don't remember Alexa's talk. We rescheduled her from earlier in the year, and so she will be our speaker for the last speaker of this year. And we will send out a new schedule as soon as we've got it finalized, as well as some potential to do some volunteer if you have the time and interest on some additional projects around science and research related to beavers that Brian and I are interested in collaborating with everybody. So I think those are all of my announcements. We will be trying, based on some feedback and getting to know Zoom better, we will be trying to actually allow people to talk and ask their questions as opposed to having to put them into the chat. So again, we really thank everybody for their patience as we work through this new format. We ask for patience again today as we try some new things. But as always, if you have a question, please feel free to put it in the chat. Brian and I will monitor them and make sure that Jesse, our speaker for today, is aware of them. All right, Jesse, are you ready?
I'm ready.
So welcome. We'd like to welcome Jesse Burgher, who's a PhD candidate at Washington State University in Vancouver, Washington. I met Jesse from reading one of his papers that his advisor had sent out related to environmental DNA in beavers, and I thought this would be a great speaker for our series. And so today he's going to talk about his current doctoral research, assessment of environmental DNA for detecting and monitoring translocated North American beaver and other applications.
Yeah,
A little extra.
ended up adding a bunch of other stuff for my dissertation because that's how it goes.
Right.
As Sandra said, my name is Jesse Burgher. I'm a PhD candidate in the POV Scott Lab here at Washington State University, Vancouver. I'm in my 5th year. I should be graduating in the spring, and I'm going to be talking about kind of our beaver-related research program that involves a lot of beaver-related restoration as well as beaver relocation research. And most of my interests kind of lie in wildlife biology, molecular ecology, and restoration ecology. And so that's kind of where this angle of environmental DNA came from. And before we start, I always like to kind of thank and introduce kind of the beaver-related restoration research team here at WSU. My PI is Dr. POV Scott and Dr. Goldberg. There's me in the middle, as well as Alex Duke. She's a lab technician in the Goldberg lab. She was a master's student who helped with some of this research, as well as Juliana Halza, who's another PhD student in my lab working in these systems. So we're going to be talking about beaver-related restoration. This is a suite of process-based restoration tools that has been gaining a lot of traction across the Western U.S., really trying to work with or mimic beaver engineering to achieve some of these restoration goals. And this is kind of inclusive of beaver relocation or reintroductions, as well as the construction of beaver dam analogs. And as you beavers are these kind of really unique organisms on the landscape. They are these quintessential ecosystem engineers. They have drastic hydrologic, geomorphologic, and ecological impacts in freshwater ecosystems. The most iconic of which is the dam, which creates these complex wetland systems that can sustain biodiversity in the face of climate change and some of the changes that we're seeing. And beaver relocation and reintroductions really has been a tool that has been increasingly implemented. I kind of use Washington as the example because that's where I am. It's been dubbed this sort of sizzling hotbed of castor relocation, and you can see kind of this variety of headlines highlighting how much interest there is in this, which kind of culminated in the development of this beaver relocation pilot program through WDFW, the Washington Department of Fish and Wildlife, that started in 2019. I believe it is no longer a pilot and it is a full-blown program at this point. And this is really increasing across kind of other states in the West. We've got, new beaver management plans kind of incorporating their value on the landscape. And this is Colorado and as well as California starting to incorporate this type of restoration into their sort of state wildlife management plans. On the other side of the BRR coin is the beaver dam analog. So these are also really increasing in implementation across the Western US. These are man-made structures that are meant to mimic beaver dams. So they consist typically of sort of these pounded posts across the stream that are woven with willow and then packed with sediment with the hopes of creating some of that same sort of ecosystem engineering abilities of the beaver. These are kind of standardized now through the Utah State University Restoration Consortium, Dr. Joe Wheaton and his restoration company, Anna Branch Solutions. These are going in the hundreds to thousands across the Western US. But they're pretty new on the landscape, and we still really don't know a lot about what's going on with kind of the outcomes of these structures, as well as with the beaver relocations. And so we've used eDNA to kind of hope to help improve beaver-related restoration through kind of a couple bodies of work. First was using eDNA to help assess beaver occupancy at these relocation sites to better understand distributions on the landscape, as well as some of these movement patterns. And there's kind of a subset to this is using eDNA to help understand biological outcomes of beaver-related restoration. And then we've got the second body of work that I'm going to talk about a bit, which is using environmental DNA and these molecular methods to assess risks of moving invasive species and aquatic pathogens. But first, I'm going to talk a little bit about environmental DNA. So environmental DNA refers to DNA that we can collect and extract from environmental samples. This is often soil, water, air, ice. Feces is often lumped in with this as well. We can collect this DNA without needing to isolate or capture any of our target organisms. So environmental DNA is often this complex mixture of mitochondrial and nuclear DNA from a bunch of organisms that we isolate from one of these samples. We then preserve it. We extract the DNA, and then we take it into the lab for these kind of downstream processes. But the DNA in the environment is subjected to forces of decay. There are kind of forces of absorption into different substrates. There's resuspension in aquatic environments. And there is sort of distributional or dispersion kind of forces that act on it. All this to say that eDNA is often very patchily distributed in the environment, and it's often decayed, short fragments of DNA that can make kind of the process challenging. I typically work with aquatic eDNA, and so that's what I'll be talking about today mostly, but this is just a little kind of cartoon to highlight sort of the different types of samples you can collect and that process of going from the field to the lab. And one thing I want to highlight is this assumption that aquatic eDNA detections reflect the contemporary presence of the organism you're looking for. And this is kind of where a lot of the strength of eDNA comes from, aquatic eDNA. But I'll kind of highlight how it's tested in the Beaver system a little bit. I have a kind of complex schematic of this process that I'm going to walk through relatively quickly, but if anybody has questions about specific steps, we can ask and talk about those later. But the idea from presenting this is to kind of show you these two different divergent analysis pathways and sort of the types of questions you can answer with those. And then also just to highlight the level of complexity that goes into this type of analysis. So we basically collect water samples in the field. We always start sort of the same way. We in our lab tend to use the Smith Root eDNA system. That's a electrofishing technology company that develops a bunch of eDNA equipment. So we've got these sort of self-contained filter units, and we typically filter our water on site. So the idea is you're filtering water through a filter with very fine pores, collecting the DNA from that, preserving the filter, and extracting DNA from that. There's a lot of ways you can do this. You can collect grab samples, put those on ice, bring them back to the lab and filter them. But we've found this system to be really effective. After you have preserved that filter, you take it to the lab and you extract DNA from that filter. There are many different ways that you can do this. We tend towards the Karen Goldberg's protocol, which is sort of standardized for environmental DNA extraction from filters. But again, there's kind of a lot of different ways that you can go at this step. And after this step is where you get these two sort of divergent analysis techniques. So the first is this quantitative PCR for species specific detection. And that's going to be what I'm going to talk about with the beavers a lot. But essentially, you take the DNA extract, you have a species-specific primer. It's a DNA template that is unique to the species you're interested in. You use that with your DNA extract through quantitative PCR to identify and amplify that DNA in the sample. Then you go through a bunch of analysis stuff where you do QA, QC, rerunning consistencies. Essentially, you get detection at the end. So you can estimate detection probabilities, presence, absence, and DNA quantity. The other pathway is called metabarcoding, and this is more used for community assessment. And instead of using a species-specific primer, you use a more sort of generic barcode. The idea behind the barcode is to kind of thread this needle so that it's specific to the taxonomic group you're interested in, but conserved enough across that diversity that it amplifies the DNA from everyone in that group. So you're looking at specific mitochondrial DNA gene regions that are unique to all fish, or specific to all fish, won't amplify mammals, but will amplify all fish. You attach specific codes to the DNA sequences from each sample, then you pool everything, sequence it, then you pull that back out to the different samples. You get a massive file with a bunch of DNA sequences. And then you compare that to known sequences. So you compare that to GeneBank, and then you get sort of full community diversity and potentially relative abundance. All this to say, each step involves these decisions and testing to ensure that your eDNA data is addressing management and research questions. And you kind of have these two ways that you can use this tool to address questions and management concerns. And across systems, environmental DNA has been shown to be this really good kind of complementary tool to a lot of traditional monitoring methods. It's often has higher sensitivity than traditional methods. It's non-invasive, which is really nice. You don't need complex permits. You can go out and just pump water. You don't have to, get fish permits to go handle fish and things like that. And it can be pretty easily standardized across systems. You can create these protocols and then kind of put them in place across the landscape. And it does often have reduced cost and labor compared to traditional methods. So that can vary by system. And specifically in Beaver systems, a lot of people are using some of these more traditional methods, especially in relocations. You're kind of putting out wildlife cameras, or using pit tags to try to or beaver sign surveys to try to understand occupancy or kind of relocation success. But those are pretty geographically fixed. And if the beavers kind of leave, it's hard to know that they left. And so I think eDNA, when we started this work, hadn't really been used in aquatic mammal systems very widely. And this was a sensitive tool that we thought we could apply to this question. And so we were interested in assessing how we could use environmental DNA to sort of manage relocations, understand relocations, and beaver distributions on the landscape. So we have two studies that kind of complement the start of using eDNA in these systems. The first was just to compare eDNA detections to beaver activity sign at sites to see if active sign, how detection probabilities correlated with the level of activity we would assume from that activity sign surveys. And the second was to actually do this more spatially explicit assessment of eDNA detections with beaver occupancy and movement site use patterns. And we worked with these 5 different groups across Washington who were either doing beaver relocations or just working in those systems and knew where beaver sites were to kind of implement these 2 studies. So this first study was actually run by Alex Duke. She was the primary on this study, but I had collected about half the data and helped with the manuscript and stuff. This was really looking at environmental DNA versus activity signs, and this was kind of the first time we were using it in these systems. So we had some basic questions like how consistently were we detecting beavers with eDNA, and how that detection probability related to that assessment of beaver activity. Either there was no sign, there was old sign, or there was fresh sign. And we also tried out a few different sampling methods to see what was most efficient for detecting them. So this is that Smith Root backpack sampler, super fancy and expensive and nice. We tried 2 different methods with that, either just taking point samples or actually walking a transect. And then we also just tried out a hand pump method. far more cost efficient in terms of the materials you need to get going. This study specifically involved Cascade Forest Conservancy, the Cowlitz Indian Tribe, and the Methow Beaver Project. They helped us identify occupied sites where they had done relocations but were sort of uncertain of the occupancy status, and then sites that they were targeting for future beaver relocation efforts as a kind of a proxy for habitat suitability. So we had 38 sites in these two regions, the Methow Okanagan area, and this is the kind of the Gifford Pinchot and Southwest Washington. And at each of these sites, we had this sort of central focal point, which would be kind of the release location. And we did sign surveys 600 meters upstream and downstream of that location, recording beaver sign. And at each site, we collected at least 2 eDNA samples, sometimes more. And then at a subset of those sites, we did those multiple methods. So we were looking for sites that were kind of big enough to do those transects on. And there's not really any fancy figures for the outcomes of this study, but we kind of found that eDNA complements sign surveys, but detections wasn't always related to sign. So just kind of addressing the initial questions. We did find that eDNA was pretty effective and consistent at detecting beavers. So 81% of the samples collected at those sites with fresh sign, we had detections, which is pretty good. For the second question, was it related to activity signs? It's kind of a maybe. Statistically significant higher detection probabilities with active sign, but we did have high detection rates at sites with no signs and sites with old beaver sign, what we classified as old beaver sign. And finally, we did find that those kind of point samples with the backpack sampling were the highest efficiency. But this study kind of, first test of these in the system in beaver systems. I think it is a good complementary tool for helping confirm occupancy or absence when that kind of sign is ambiguous, but it left us with a lot more questions. And this is actually in press in Northwest Naturalist right now. Simultaneously, we were looking at this study that was kind of this more spatially explicit assessment of eDNA, how those detections relate to known occupancy and sort of site use patterns. So I worked with these 3 different beaver relocation groups who were actually doing the relocation to get access to these release sites and to actually get access to the beavers. And these were kind of our basic questions in this system as well. So how reliably was eDNA able to detect beavers if we knew they were at the site? And then how those detections and those patterns of detection relate to where those beavers were within the system and how far away were we able to detect them. Because we had those detections when we didn't see any beaver signs. We were interested in if that DNA was maybe flowing through the system and we were detecting upstream beavers. And so to do this, we used beaver relocations as this nice study system where we could go out, find a release location, sample it for eDNA, confirm there was no DNA present, and then trap beavers, tag them with radio tags, and release them, track their movements, and then collect eDNA over these fixed distances kind of over time. That way we could attribute all of our DNA detections to those beavers that we were tracking. And this was kind of what that looked like. So we had 4 study sites in Washington. This was really logistically challenging. I wish I had more sites, but this is where we ended up. At each of these sites, we released 2 to 3 tagged beavers. So all these sites were negative for beaver DNA prior, and then we radio tracked and collected eDNA samples over time. So each of these little yellow triangles is where we collected an eDNA sample at each interval. And then the dots are actual beaver locations. We've got different colors for different time intervals, and the star is that release location. Collected a total of 270 eDNA samples as part of the study. So we had a lot of data for this study. And this is the Snowy Creek release site near Leavenworth. This is kind of the best example of this system because they actually built dams and stayed here, and their movements were a lot less than some of the other sites. And so what we did is we released them, and then we tracked at different intervals, different intensity for the different time intervals. So at the first day, I tracked them overnight. So I got locations every hour for every beaver. And then in that morning, I collected eDNA samples at the most downstream location, so down here. Then during the first week, you can see those other locations added in pink here. I relocated the 3 beavers every day. And then at the end of the week, I collected eDNA samples across the whole site. And then every month after that, I relocated the beavers and collected samples across the whole site. And this is what I mean by the site being the best kind of example, because they built dams and they stayed right here. So I tracked these beavers for 3 months before the snow kind of limited my access to this site. And what we found was that eDNA detected those beavers really rapidly over long distances. So within 24 hours after release in these kind of clean systems, we detected beaver DNA at the most downstream locations, which was about 2.4 stream kilometers downstream within that time frame. So within 24 hours of release. So most downstream beaver locations to most downstream eDNA sampling location. Looking at some of the more longer term data that kind of more addresses the reliability and consistency. So when beavers were upstream of where we collected an eDNA sample, we had really high detection probabilities. So if a beaver was here and your stream's flowing sort of this way, and you collected an eDNA sample here, we had 99% probabilities of detecting upstream beavers, regardless of the distance. And so from our modeling, our model estimates show a 99% probability of detection. You can see the raw data in these open circles and our 95% confidence intervals. One of the interesting things to highlight is this, probability of detection when beavers were downstream. This figure is from our long-term data. And so I think that this uncertainty sort of reflects the coarseness of our radio tracking at that interval. So I think, beavers were probably moving a little bit across some of these locations that wasn't really captured by our radio tracking in the longer term. But we did have that first week really spatially explicit data where we knew where they were every day. And it's a very consistent pattern with this data. We also looked at DNA quantity. So through eDNA, you can look at the quantity of DNA in a sample. And we wanted to look at how that quantity of DNA was related to how far away you were from the beavers. And what we found was this really nice pattern of decreasing DNA quantity with sort of increasing distance between those beavers and where you collect a sample. And what we found is for every 1,000 meters of stream kilometer that you add between where that beaver was and where you collected the sample, you had a 61% decrease in the quantity of DNA. And this to us sort of indicated that if you collect sort of samples in this interval design along a stream, and you see that DNA quantity is increasing in those samples, you can get an idea of where those beavers might be within the system and really increase the spatial inference of eDNA kind of longitudinally across these streams. And so I think some of the takeaways from this study, I think eDNA is very effective for detecting beavers. So for reliability, it's really rapid and really reliable. We could detect them really quickly over long distances. How those detections relate to locations, we had these really consistent positive detections of upstream beavers, and we had that nice pattern with DNA quantities that decreases with your increasing distance from established beavers. So I think you can get a lot of information about where they might be from samples, from eDNA samples. And we just really had these nice consistent positive detections over these long stream distances. We had 2 to 3 stream kilometers was the furthest distance that we were sampling. But we had really consistent detections over those distances when we knew they were upstream. The one thing I didn't talk about much was the eDNA signal persistence, which is something I mentioned in the beginning. We didn't have a design in this study for really assessing this robustly, but we did have instances where beavers abandoned the sites. We're pretty sure abandoned the sites. We had a lot of drop transmitters, and we had a lot of eDNA signal persistence. In some cases, more than 30 days, especially where beavers built structures. So I think this is kind of the main piece that we need to do some more research in, is to understand how long that signal might persist so that we don't consider detection. So we're not considering abandoned sites as positive occupancy. And I think this is unique to this system because they deposit so much DNA and they engineer so heavily that there's a lot of residual DNA in the system. But again, I'm just going to kind of highlight some potential management applications. So I think the eDNA is really rapid and reliable for detecting beaver occupancy over these kind of long stream distances and can provide some spatial inference on site use. And One of the things I like to highlight is how sensitive it was. So, we had these really high detection probabilities. It was super sensitive for detecting. We could detect 5 copies of DNA estimated in a sample. So if you have negatives, it really strongly suggests that there aren't beavers present within that system or upstream of where you collected the sample. And I think that can be useful for targeting relocations. I also think it can be useful for inventorying occupancy at some larger scales and in these hard to access areas. So for example, this is a Huck 12 watershed in Southwest Washington. It's kind of a headwaters watershed where you might be interested in sort of putting beavers in maybe some of these more headwater wetlands to try to keep that water on the landscape. It's pretty rugged. It could be hard to hike this whole thing. And so if you collected, eDNA samples across the system at like 5 locations, I think you'd have a really good idea of which tributaries and where those beavers might be within the system. And this, as we mentioned, was published in Animal Conservation this summer. So there's that citation. And I'm going to move on to some other stuff. But if anybody wants to ask questions about this work, we could do that now, or we could wait till the end, whatever people think.
Do people have questions right now, or we can keep going and you can hold them to the end?
Yeah, why don't we just keep going?
Let's keep going.
Okay. So next, I'm going to talk a little bit about sort of how we think about using eDNA to understand kind of biological outcomes of beaver-related restoration. This is a project that is in process, and so it's not going to be fully complete, but this is kind of going down that meta barcoding pathway where we're thinking about community assessment and responses to environmental change. And so in this study, we're really trying to focus on how the installation of beaver dam analogs impact these stream macroinvertebrate communities. A lot of these things are going in for fish communities, and this is kind of the base food for that. So that's kind of the motivation behind this. Also, sort of the traditional collect, sort, and ID methods for macroinvertebrates are really labor intensive and rely on specialists, and it takes a lot of time. And there's been a lot of literature highlighting the promise for metabarcoding for detecting community change and being sensitive in these systems, but it's pretty complicated. So we have a system in north central Washington where we have these unrestored reference streams, and these are upstream of these beaver dam analog restored sites. So we've got these paired unrestored and restored systems. And then we have these natural beaver systems that are close by. And so this is the map of this site. So this is kind of the Methow Valley and the Okanagan Highlands. And we've got 11 beaver dam analog restoration sites with upstream pairs, and then 7 beaver sites. And in those systems, we collected 2 to 3 eDNA samples kind of across their stream reach. We extracted DNA, and these samples are at a commercial lab, Jonah Ventures, who are going to do the metabarcoding sequencing. So those are just kind of to highlight how we're thinking about eDNA and metabarcoding and community assessment with these tools. But more information to come on this study as we get further down the road with it. And I was going to talk a little bit about this second kind of body of work, which is maybe a little bit outside the scope of the original title, but I think it's interesting, and it highlights another application of eDNA sort of in these systems. And that's using it to assess some of the risks and exposures of beavers to aquatic invasive species and aquatic pathogens. So this has been a concern of a lot of the agencies and folks who are managing beaver relocations. And we're in a lot of these systems, we're trapping beavers at low elevations in areas where you would expect to have, high prevalence of invasive species, potentially pathogens, and we're moving them up in watersheds to this more kind of pristine or quote unquote pristine habitat, which creates this pathway for co-dispersal. Mammals have been implicated in the movement of invasive species and pathogens in their fur, but it hasn't been assessed for beavers. And so we're looking at some kind of basic questions of if they're exposed, the prevalence of that exposure, if any of some important pathogens can actually grow and maintain viability in their pelts, and maybe how we could neutralize some of those. And so we've got kind of 2 areas of study in this system. First, just looking at that prevalence of exposure, where we're actually trying to detect some invasive species and pathogens on the beavers during relocation. And the second is this kind of lab system where we do these proof of concept experiments. And this is specifically with amphibian chytrid fungus, looking at how it could grow and transfer and how we could potentially neutralize that on beaver pelts. And this was originally funded by Washington Department of Fish and Wildlife. We've got some ongoing work with them, but it also was a concern of Wyoming Game and Fish, their state herpetologist, and Colorado Parks and Wildlife also funded some work specifically in the chytrid system because they're trying to create habitat for critically endangered amphibians that are susceptible. So for the kind of sampling of beavers, we had this system where we were sampling beavers throughout their period of captivity, kind of longitudinally. So we would sample their capture location, and then we had two types of sort of samples from the beavers in the relocation system. So we'd scrub their pelts with a sterile gauze, suspend that in water, and then filter that water to detect any of the DNA for those pathogens. And we would also sample their enclosure water during captivity and then an outgoing scrub as well. And we worked with these for beaver relocation groups to help collect these samples, like made some kits and some protocols and sent them out to them to help collect the samples. Because the diversity of these invasive species and pathogens is pretty broad, we don't have a meta barcode or a barcode that would work. So this is all sort of species-specific detections using quantitative PCR. And so we limited ourselves to 4 target species. We were looking at ranavirus, which is a viral pathogen of amphibians and fish. We were looking at BD, so Batrachochytrium dendrobatitis, which is a fungal pathogen of amphibians. We were looking for New Zealand mud snail, which is an invasive mollusk. And we were also looking for Flavobacterium psychrophilium. This is a bacterial pathogen of salmonids, and this is kind of our control. This is purported to be really widespread in Washington. So we wanted to include something that kind of confirmed that the methods were going to work. And so this is a hatchery facility where they hold beavers. This is in Leavenworth. Just an idea of what that looks like and how we sample. It's kind of sample the beavers. So we hold them in these beaver bags, and we can scrub their little butts and get the samples from their pelts. We had 102 samples. This figure only shows samples collected in the facilities, not the release locations. And about 40% of those samples were positive for one of our targets. We had one detection of amphibian chytrid fungus, and the majority of them were for that Flavobacterium psychrophilium, which is sort of what we expected because this is our kind of positive control, right? But what is interesting is that we found a lot of these beavers ended up being exposed to this pathogen or this bacteria in those hatchery facilities. So their intake scrubs were negative, but their facility samples and their outgoing scrubs were positive. So they've kind of highlighted that these hatchery facilities are this kind of potential source of exposure to primarily fish pathogens. However, this chytrid positive is concerning. And so there have been some regional pauses on relocations in Washington and Southwest Washington, and we're kind of continuing to work with WDFW, as well as there's been some collaboration with the National Wildlife Research Center to do some additional sampling of beavers to look at these questions of exposure and prevalence. And there's plans to do further sampling, kind of expanding that out pending some pilot data that we're working on now. This is a transmitter that we attached with Stacy Robinson in the National Wildlife Research Center locally, and we did some scrub samples of these beavers. So it's just kind of a cool picture. So that's the ongoing work in exposure prevalence. I want to talk a little bit about these lab experiments because there's some interesting outcomes here as well. So these are these proof of concept experiments where we're looking at how the pathogen can transfer on beaver pelts. So we run a amphibian pathogen culturing program in our lab, and we use these proof of concept experiments to kind of balance realistic properties of the biological system and kind of ideal growth conditions for the fungus so we can observe some of these dynamics. And what this looks like is taking sections of beaver pelts, sterilizing them. We harvest and add the infectious particles of that amphibian chytrid fungus, and then we incubate that and then transfer it to kind of these ideal growth system and growth conditions to observe how well it transfers and how long it maintains viability on beaver pelts. And in some of our sort of initial experiments, we found that about 2/3 of those pelts were transferring viable BD somewhere in the 4 to 14 days after that initial exposure, which is a pretty high transfer rate. And so we got some interest from partners to kind of look at some ways that we could neutralize this on the beaver pelts. This was funded by Wyoming Game and Fish and Colorado Parks and Wildlife. So it's the same setup, except we add these neutralization treatments before we transfer. And these are things that we could apply to live beavers. So we were looking at doing a lot of rinsing, so changing their water with clean water every day, which is going to be happening in relocation settings already. We looked at different lengths of drying. The chytrid fungus is susceptible to desiccation, so we tried 4.5 hours of drying and 9.5 hours of drying. And we also tried some salt solution applications because chytrid is susceptible to salt. So we did a 2.5% saturation and a 5% saturation. And we kind of corroborated our early experiments. We saw a lot of BD transfer and growth in about 2/3 of these different replicates. And we were able to submit some of these hairs to the scanning electron microscopy facilities here at WSU. And these are some of the first images of chytrid actually adhering to mammal hairs. And you can see these complex mats of the fungal structures, including reproductive structures, attached to beaver hair. So a bit concerning. But we did find that drying for at least 9 to 10 hours really significantly reduced by at least half the growth, the probability of transfer and growth in these different replicates. But so I think this is going to be potentially implemented in some of these states where we can start with this 10-hour drying period to help. This was the ideal growth scenario. So in the field, this is likely to work even better. And so I think this is a step we can take to start thinking about this problem, and actually pretty simple to kind of help manage it. I think there's more testing that could be done. It would be nice to find something that gives us 100% certainty that we're not moving it. And then again, just kind of highlight that we're doing some ongoing work in this system, and hopefully we'll have some more robust recommendations sometime in the future. Okay, that was a lot. So take-home messages kind of across this. I think environmental DNA is and can be very useful in beaver and beaver-related restoration systems. I think there's a lot of promise for understanding relocation success and distributions and occupancy at a watershed scale using these tools. I think there's potential to rapidly assess biological outcomes of beaver-related restoration using these tools. And I think that there's ways we can use them to aid in surveillance and monitoring risk factors in animal relocation systems. But I think there's a lot of complexity in design decisions and processing pipelines that need a bit more research attention. By the way, this is a dam from that Snowy Creek site. So these relocated beavers built this really cool dam, and we're living in a bank right under this tree. And with that, I can take any questions. I'd just like to thank my funders and project partners for helping with all this work and making the work so meaningful and enjoyable.
Thank you, Jesse, for a really interesting talk, and especially being able to touch on the multiple ways that you and your group are using environmental DNA to address different questions that I think this group is really interested in related to beavers. We have a couple of questions. Let me do, I don't know which one to do first. Hang on. Okay, so we'll start with the shorter, not necessarily easier, but shorter to say. Danielle Garneau it was, I don't know who BF is, but was the salt treatment successful?
So no, the long drying was the only one that had any treatment effect in that system. And I think from a physiological standpoint, I mean, 10-hour drying is not, the beaver's not going to be super happy, but and from my perspective, I'd rather do that than saturate a beaver down with salt and risk salt ingestion toxicity. So, but we did consult with state wildlife vets on these different treatments, and they were all within sort of like an IACUC framework. So
another question from Jim Shepard. So Jim has been monitoring a watershed where he has more than 430 streams with 77 confirmed to be beaver populations. And
Okay,
most of these confirmations are actually up in the tributaries that don't regularly connect to the main stem.
interesting.
So for your eDNA work, looking at where you said that one application could be for surveying hard to reach locations. Um, Within that kind of system, instead of sampling the main stem, would you be able to use that, still use it, but looking at the lower end of these creeks, even if I think the implication is that perhaps they dry at some times of the year?
Yeah, that's a good question. I think you'll have even better inference if you're sampling in the tributaries. So from our research, our best sort of level of understanding is that you get detections in about 2 to 3 stream kilometers. So if you want to sample those tributaries, you'll have a good idea of occupancy of a tributary. Once you sort of get into the main stem, you're going to get DNA mixing from all those different systems or all those different tributaries that are flowing from above. And so it's harder to understand which tributary might be occupied if you're taking samples from the main stem. So I think sampling in tributaries is probably how you would want to target it.
Oh, Sandra, we can't hear you.
Okay, let me try that again.
No worries.
I was just saying, I think we have the ability to unmute you. If you would like to say your question instead of typing it, we can do either. We're going to try either. So we will see what we can do. All right. Let's see. We still have time for questions. And so as I let
Oh
people
yeah, that's fast.
take an opportunity to put their thoughts together and perhaps type them out, um, I had a question.
Yes.
So with your work with looking at potential disease spread, is your sense that this is mostly a concern in relocation as opposed to natural populations moving across the landscape and becoming, say, beavers from a BD prevalent landscape moving into a low BD landscape?
That is a good question, and perhaps one more for the people in the management side of things. But from our work thus far, it's been more a concern with the relocation because we're really doing these kind of rapid, larger scale movements in that case where it's, with the actual animal populations expanding might be a little slower. Maybe. I mean, beavers sometimes move pretty far, but I think it would be more of a natural progression. But I don't know. Sean Belling's on this. She might be able to weigh in on.
Does anybody want to unmute Sean and see if she can make a comment? I don't know if I can maybe manage all of those buttons at one time. Let's see if oh, there's Sean. Ask to unmute.
I just unmuted her if she wants.
Hi, yeah. I'm sorry. Can you repeat the question?
So
Sorry
the
to put you on the spot, Sean.
yeah,
Oh,
so
good.
Jesse's passing the question to you, which was the work with the relocation is really interesting, and I see its relevance. And I was wondering though, is there also concern about beavers transferring invasive diseases across the landscape as they move into new areas naturally, as opposed to human relocation?
And that is a fantastic question. We're so terrified about diseases moving the ones we are translocating. We don't have time to worry about what's happening naturally. We barely have the funds to be able to track what's happening with the relocation program. So, yes, we would love to learn more about that. It is not even on our radar yet. It just keeps us up at night, and there's nothing that we can learn about it quite yet. But hopefully, now that the relocation program has become a permanent program, we're hoping that more funds will be able to come our way so we can send more money to Jesse and his team and learn more about this on all levels. No promises, but that's the
Oh
hope.
yeah. Yes, we're doing more of the sampling right now on a very, very shoestring budget that we're making work, which is often how it is. But
Yes, exactly.
So on a practical side, I'll just keep asking questions until another one comes
yeah,
in.
that's
So
fine.
On a practical side, you just mentioned budget. And so how, what is the financial, just kind of ballpark, not little, you don't have to nickel and dime all the items, but if you think about if people wanted to start using eDNA as part of their monitoring program, what kind of investment do they need to make? Both in
Yep,
terms of collecting the sample, but also that downstream side of the sequencing and the analysis of the sequences. And how automated
sure.
does that become?
So it's a good question and one that
Oh,
gets asked a lot. I think the Smith Root, the company that we kind of work with their equipment a lot, has these citizen science samplers that are pretty cheap, sub 2000. I mean, you can go cheaper than that too, but they're really kind of dummy proof in a lot of ways, and it's a nice pump. So initial equipment could be less than that. Like those hand pump setups can work for about $100. The filters are about $16 a piece. And then downstream lab stuff is about $30 to $50 for species specific qPCR. For the metabar coding, it's closer to $100 a sample commercially. It is starting to be automated. This is my first time working with a commercial company with some of my metabar coding stuff, and it's challenging to say the least because there's not a lot of it's kind of like they just have the way they do it and they kind of do it the same for everything, which isn't really how it works. You kind of have to tweak different steps for metabar coding. So I wouldn't necessarily go that route in a monitoring program yet. For species specific detections, for sure, that's pretty bulletproof. I think for metabar coding, it's a little more complicated. But yeah, I think for species specific, you're probably looking at $60 a sample. For metabar coding, maybe about 100.
and
So
yeah, and
yeah,
Anne McMillan had a similar question. I think you've mostly answered it, but I do want to say it just to make sure there's any nuance I didn't capture as well. So again, looking at eDNA costs per sample, which you've alluded to there. Um, so there are a lot of creeks that she's interested in, but not permitted to enter. And so um, there was a question. Oh, she says yes. You did answer it. So I won't say it then.
okay,
If
cool.
you're good, Anne.
Yeah, I mean, I think that's one of the things that the transport distance that we've found can help overcome, right? So if you want to sample a creek, but you can't access a specific area, you could go a couple kilometers downstream, maybe where you can take a sample there, and then you could potentially understand what's upstream from that, um, which I think could be useful. The other kind of nice thing about an eDNA sample, it doesn't take the whole filter to actually do the processing for one project. So you have this kind of record that you could come back to, and then you could look for another species. So you could it kind of creates this more permanent record of DNA that has a lot of utility, I think, over time as well, as long as it's preserved well.
Well, I think another question that you touched on in the first part of your presentation, but if you could maybe expand a bit, is this question of the fact that DNA stays in the system. And
Yeah.
so how does that become a potential confounding factor of doing just basic monitoring versus the type of work that you're doing, which is targeting around relocations?
It's a good question. I think it is complicated. One thing that I noticed from some of our data, this isn't, we didn't have enough of it to do a robust statistical analysis, but the difference between detections with an occupied site and with a non-occupied site were pretty different. So when you had an occupied site, all of our replicates were positive. All of the downstream samples were positive. When we didn't think it was occupied and we were thinking we were detecting residual DNA, it was a lot patchier. Like 50% of the replicants were positive. We still had positive detections, but it was a lot different than when they were there. The other thing is the quantity of DNA in those samples was about uh, fivefold less. It was much, much less DNA quantity. So if you're able to collect sort of more than one sample in more than one place, then you might get an idea, even from a positive detection, if you think that's actually occupied or not. Unfortunately, I don't have enough data to actually put that in the paper. It's too speculative, but like those are the patterns we see. And there were some sites that we were intending to do this work in where we had detections that we think was residual because it would be like one replicate of a sample with a really low level detection. We didn't want to use that for the study, but those were previously occupied sites. We're no longer occupied. It was just a residual detection. That occurs more often in a beaver pond than in the stream as well. So if you're in sort of like a sediment-rich pond, DNA binds to sediment and can stay in a system longer that way. If you're in sort of flowing water, the chances of that occurring or what is occurring there is probably different than in a pond, say. Like this pond in this picture here, if you stirred up the sediment or if you accidentally sucked some of that sediment into your sample, you'll probably detect beavers. I don't think it's occupied. And if you were just sipping off the top, you might not. But that's kind of one of the dynamics that can happen with eDNA.
Anne has a question, and Anne, I unmuted you, I think, so you should be able to ask it.
I'm sorry, I must have my hand raised accidentally.
Oh, okay, so you're good?
I guess I do. How do you tell, are you basing your,
Nope.
We lost you.
how do you make the call that the beaver used to be there, but they're not there now? Is that where you're doubling up your sampling with your observations?
So in this system, we had the transmitters on them. So we kind of knew when they were gone. I think in an unknown system, it's more questionable, right? And that's one of the things I think we still need to address with this tool, sort of in this system, with probably another project, which I have thoughts about, but no money to do.
Thank you.
Yeah.
So a question related with especially the relocations. Are you able to, is there a method with the DNA samples that you have that if you had the DNA from the beavers, would you be able to say like which beaver is somewhere?
I get this question all the time. Not yet.
Not yet.
There is some work ongoing with detecting individuals with DNA sequences. There is some polar bear work where they're able to do this with licks. So they put out these kind of baited licks and they collect the DNA from that and they can actually track individuals. There's been some eDNA work. It's not finished yet, but there's some stuff. Karen has a data set looking at pond turtles and trying to track individual turtles using eDNA. It's complicated by the fact that we're collecting these really short DNA fragments. A lot of the times the eDNA is like 200 base pairs. And so it's really hard to get enough information from that to track that to an individual. So that's kind of what we're trying to overcome in these systems. And we'll see what happens with more research, but yeah, it's not there yet for sure. There's another common question with eDNA is, can we estimate abundance from the DNA quantity? And there are some relationships there, more animals can be related to increased quantity of DNA, but it's really system and organism specific, and it can be really time specific. Like, for example, reproducing organisms are going to produce a lot more DNA than more non-reproducing organisms. There's also these phenomenons with super shedders, which we don't really fully understand, where there may be like one organism that's shedding far more DNA than the other ones of the same species. So that's kind of something that is in development, but it seems like every time a paper comes out about that, it's like system specific. And so it's kind of we haven't been able to identify a pattern across systems and species for that at this point.
We're coming up close to the hour, and I want to be respectful of people's time. Are there questions? I've kind of dominated the question period because this is a topic that I'm very interested in, and I do a lot of, not a lot, but also some eDNA work associated with biomonitoring. So Jesse and I've had other conversations around this topic, and so he knows that I'm very interested in it.
Yes.
But I wanted to give other people a last opportunity to ask questions. I did put Jesse's contact information, his email, into the chat. And I would remind people that Avery will make this webinar available on the Beaver Institute YouTube channel. It'll take her a few days before it's posted, but you will also be able to access it there if you wanted to reach out to Jesse and ask him more specific questions.
Yeah, please feel free to email me if you have any questions or you want to talk about projects or anything.
All right.
Awesome.
I think we're good. So let's all thank Jesse for presenting today. Really appreciate you giving your time and sharing your research with the in the speaker series. A reminder that in December we have Alexa Whipple, who will be talking about something I assume associated with the Meetow Beaver project. I have her title in front of me. I apologize to Alexa. And as always, if you have questions about being involved with Beaver Institute or the Science and Research Group, feel free to reach out to me or to Brian or to Avery. And Brian and I will be in contact sometime over the next month-ish with the updated schedule for next year, as well as some opportunities to do some additional work around science and research. So thank you, everyone.
Thank you all. Definitely big plug for Alexa. She helped us with a lot of this work, and she's a very infectious speaker, so don't miss that one.
Great.
Thank you.
Cool.